I will remind you to wear your hat and your sunscreen.
It's true. I've seen it. Hello and welcome back to Leaf, a LI-COR plant physiology podcast where we are covering all of the chapters to
A Hitchhiker's Guide to Gas Exchange.
Thank you, Elizabeth. A Hitchhiker's Guide to Gas Exchange, a book written by Jason Hupp and Dr. Richard Vath. So this is... Which episode is this? This is
Episode 7.
This is 7. And that's right. Not only do we have new epic, heroic intro music, which was selected because I do, in fact, believe this next guest and her group to be heroes. So without further ado, let's go ahead and welcome my very esteemed guest here, Elizabeth Gordon. Hello and welcome to the podcast.
Thank you.
If you're wondering, yes, it is in fact that Elizabeth. If you have called with any particular issue, concern, or if you were like me and maybe called in tears asking for help while you were in the field, this is in fact her. And so I'm just gonna kind of hand the mic over here to Elizabeth to introduce herself.
Sure. Well, yes, my name is Elizabeth Gordon. I am the Elizabeth, if you've ever called LI-COR. And I say that because I'm the only Elizabeth who works at LI-COR. So sometimes people do say that. So yes, I'm the Elizabeth. There's only one of me.
If there was two, she would still be the Elizabeth. Go ahead.
Thank you. My background is in plant ecophysiology. I came out of a carbon source sink relationships lab at Texas Tech University. And I got here because I finished my master's degree, had been using the LI-6400 as a technician and a graduate student. And then I finished my master's degree and I needed a job. So I applied to various places, and one of the places I applied to was LI-COR. They had an opening at that time for someone to do technical support. I'd been taking care of the LI-6400s and all the other LI-COR products in our lab for a few years, and I thought I could do that job. I'm good at troubleshooting, I'm good at problem solving, I know these instruments. So I applied, they called me, I interviewed, and I have been here for over 17 years now.
It's been 17 years.
17 years, yep. Yeah, and my function is, as Seton implied, to provide technical support. So if you have a problem, a question, a concern about your instrument, you can contact us by phone or by email, and I will do my very best to help you. Whether it's something simple, like what is this part number, or something complicated, like how do I set up my experiment. I can't always help with the how do I set up my experiment. That's kind of up to you, but that's kind of what this book is about, is to try to help you understand the things I would guide you towards while not actually telling you exactly how to do it, because that's your job.
Well, I think you kind of just summed up the whole book, and I'll just go. I think, Elizabeth, I think you should just take over this podcast series because you actually summed it up better than I ever have. You know, I've had the pleasure of knowing Elizabeth now for, golly, about 10 years?
I think it's been about 10 years, yeah. I started working with you when you were a graduate student.
Yeah.
At K-State.
I was a wee baby.
Yes, and then, yeah, we interacted at Fizz Fest, and we've met each other at ESA a couple of times before you started working here, and now we work together.
It is a lot of fun to have known someone at different stages of a career.
Oh, and it's also one of my favorite things.
Right, from external and now kind of this internal, and you're like, oh my gosh, why did we hire you? Well, maybe this is one-sided. I've had a lot of fun interaction with you, Elizabeth. We have an incredible science
amazing.
and support group.
They're so good. You know, people say things like, people give me a lot of credit, and I appreciate that, but I am not the only person. I am definitely part of a larger team that really, really wants to help you do good science and get good results from your instruments. And I am not a one-man show, one-woman show. And so, yeah, anybody who says, oh my gosh, it's Elizabeth, she's so great, I appreciate that, but I have to give compliments to my entire support team. They're so good at what they do.
I typically called to get some help. have
Mhm.
something have something resolved. But if everyone and anyone that's listening to this, could you take out just a little bit of time to like send a thank you email, or not necessarily a call, but just reach out and say thanks, because we make some pretty great stuff and we've got some really great scientists on staff. And sometimes it's nice to hear some positivity.
I
From
have a
science.
collection of handwritten thank you notes
Mm.
on the wall of my cube because, yeah, it is good to know that people appreciate how hard the support team works. We are well aware that when you call customer support for other things, you don't always get that same experience, but we want you to. You purchased a LI-COR piece of equipment, whether it's a light sensor or eddy covariance or a soil flux system or a photosynthesis system. You purchased that. And the joke I make, and everyone who's been in a training with me will have heard this already, is that when you buy the LI-COR equipment, you don't just get the equipment, you also get the entire support team, but we don't come in the same box.
We'll get back to the podcast right after a word from our sponsor. In a world where science can't wait, one researcher is running out of time. Come on, come on. Why won't you work? And when all hope is lost, a team assembles. LI-COR Science and Support is here to help you troubleshoot and get out of a jam. They're more than support. They're heroes. Oh, thank you. My experiment. It worked. I guess I could have just read the manual. LI-COR Science and Support. Coming soon to a lab near you.
As you said about, following people, that is one of the things I like best about this job. I have followed students from graduate school to a postdoc to their first faculty position. I've talked to them about getting married, about having children. I've gotten to know people as well as researchers, and I really enjoyed that. I mean, someone calls one time, and this is one of my favorites. They called one time, and they were normally a fairly cheerful sounding person. And they called and they sounded just absolutely exhausted. And I said, "Oh my gosh, what is wrong?" And they said, "I have a 3-month-old. She doesn't like to sleep." Oh, well, that explains everything. And so I said, "Well, I hope you do get some sleep." In a recent email exchange with someone, they mentioned that they were going to be going kayaking and paddling, and I reminded them to wear their hat and their sunscreen and said, "You didn't think you were going to get, life advice along with your instrument advice, but that's what you get here. I will remind you to wear your hat and your sunscreen."
It's
If
true. I've seen it firsthand.
Yes. Yeah, I told you to go call your wife because you kept not calling your wife at the booth.
It wasn't avoidance. I was talking with the customer.
I know,
My
but
wife listens to this,
but
I'm gonna get in trouble.
yeah, but you still said 3 times, "I'm gonna go call my wife." And so finally I said, "Seton, go call your wife." And you left the booth and you did it.
It's true.
So,
Oh, I wish I could edit that out, but I don't think I will. I think I'm gonna have to leave it in. So thanks for starting this off with getting me in trouble.
you're
But
welcome.
since you are there's gonna be people that want to know a little bit more about you, and I
Sure.
hope you're okay sharing some stuff.
Yeah.
Favorite book?
I'm a reader, so trying to pick one favorite book is gonna
Yeah,
be really hard.
that's why I knew
So
this was hard.
I'm not going to. I'm going to give you a favorite author, which is Ella Montgomery. I
Okay.
love all of her various Anne of Green Gables books, Emily book, things like that. Things like that. I cannot pick just one of her books. So I'm going to say Ella Montgomery as a favorite author.
Okay. I know you've explained this to me before. You basically name almost everything, and you do this through various scientist names.
Yes, so I decided that all of the instruments that the support team takes care of would need names.
Hmm.
I decided that all of the instruments were going to be named after female scientists.
Right.
So mine is named Ada after Ada Lovelace, but we've also got Barbara McClintock or Babs. We've got Dorothy Hodgkin or Dot. We've got Emmy Noether.
Hmm.
We've got Grace Hopper. Hypatia, and I'm going to mispronounce this, Chen-Sheng Wu, who was a Taiwanese physicist.
Right.
So I actually have lists that can go down. I don't ever think we're going to get to that. But all of the LI-6800s in the support team are named after female scientists.
That-
Because I get to.
Can you narrow down who's your favorite female scientist?
That's really tough. And we actually haven't gotten to her yet. And it would be R for Rosalind Franklin. The amount of work that she did to help us isolate DNA and the lack of credit that she got for it at the time because she was just a woman is such that she is one of my favorites female scientists out there. And you know, she died early, so she didn't get the Nobel Prize because it can't be awarded posthumously.
Right.
And the reason she died early was probably all that radiation for all that X-ray crystallography.
That's probably a pretty good guess.
So, you know, this thing that helped us learn so much about our world and DNA probably contributed to her death, just like Marie Curie. Turns out radioactivity is bad for you.
If you learn anything from this episode,
Yes.
it's that.
But yeah, I'm going to go with Rosalind Franklin.
Well, so okay.
So,
Like, if for those of you who are listening and you're not sure who these women are,
Google
them. Yeah, like give it a Google. Listen to this podcast, read the chapter, and then also go Google. Getting back to the book itself,
Yeah,
it had been in the works for a while.
it has.
A couple years, I think. I don't want to ask Jason; it's a sore subject. Could you maybe tell everyone and expand on what your role was in the context of this book?
Yeah, absolutely. So I actually have an undergraduate minor in English, and I have been spelling things and editing things for Jason for the entire time that I've been working at LI-COR. So, and he'll tell you that. He'll tell you that I do.
Yeah, he doesn't shy away from it.
So when he was talking about this, maybe a year or so ago, he mentioned that he would maybe appreciate some editing. And I said, great, I'd be happy to do that. So I did not write any of the content, but I did edit it quite heavily in terms of, you know, moving things around, suggesting a change, things like that. So I did not collect any data for it. I did not write it, but I would say that I was probably one of the primary editors.
That's a lot. There's a lot of pages.
It was a lot of pages.
It's hard.
Yes.
It's hard to see right now as things are released kind of piecemeal, but
It's something like 300 pages.
It was a lot of work. It was a lot of work. So we are still in Thinking Like a Plant.
We are.
In part 2 of this book. But it's really the last one before we get into this part 3, and that is, you know, looking deeper. But today is something that I've been looking forward to, and I wish I had access to some of this information while I was a grad student and early on in my career. This chapter is all about choosing a leaf to measure. Now, I'm going to say this very sarcastically. Probably never been asked that question.
Nope.
And anyone who's used a gas exchange system has likely asked that question at one point or another, hopefully early on, and things kind of start to make sense. But without saying it depends, which is one of our favorite things that we...
I really do need a t-shirt that just says it depends. I'm just going to point to it every
Right?
time someone asks me one of these questions.
It would kind of be nice because it used to be a little annoying, right, as a user when the answer was it depends. And I think this chapter does a really good job of highlighting why that is, because there are just a ton of considerations. Well, maybe we can just walk through a few of these kind of major concepts. And
Yeah.
one of the main ones is this light availability.
Absolutely.
And canopy structure. So what are some considerations there?
Ideally, we would measure every leaf on the tree. But realistically, that is just not possible. We can't. We cannot do that logistically in reality. So what we have to do is we have to choose a leaf that is representative of the leaf characteristic we're most interested in. And light availability can have a major effect. If you want to look at shade effects, but you don't measure a leaf in the shade, you're not going to get the answer to the question. And similarly, if you're interested in sun leaves and you measure a shade leaf, you're just not going to get it. So light availability is a lot more challenging, especially in a situation where you have any kind of wind, because something might be shaded, but then it's in the sun, and then it's in the shade, and
Right.
then it's in the sun. So if you are measuring within the canopy, whether that's a canopy of a tree or a canopy of a shrub or a canopy of a tomato plant, you have to be aware of the light environment that it has had in its entire lifetime, not just in this particular moment. Because otherwise, you're not answering the question that you think you're answering, and you're not going to get the data that you think you need to get.
Putting aside the I think you touched on a little bit with turbulence and things. How does one go about that, right? So is there like, should they be pre-scouting these areas? Should they a good understanding of the study system in which you find yourself? But that is, it's a tall order, right?
It is,
Finding
yeah,
out where this, if this is a temporary light situation or if it is longer term.
right. Yeah, and what we will generally recommend is either you choose leaves that are all at the same light level as measured by your LI-190 that is on your LI-600
Very
or
helpful.
on your LI-6800. Well, it's not an LI-190 on the LI-600, but it's still a PAR center. So you're going to either check that and see what Q and out external light is and say, okay, I'm just going to try to make sure that everything's around 600, everything's around 800, everything's around 1500. Or you're going to change your light level in your LI-6800. You can't change the light level in the LI-600. It is a survey instrument, but you have the choice as to whether you always try to get the same light level, and that's the same light level when you don't move the leaf aside so that it's in the same light level. That does not count. Or you say, I'm going to change it every time. Whether you want to do it one way or the other is completely up to you. But one thing you do have to be aware of is that plants respond to a change in light. They can sometimes respond very slowly, especially if you increase the light level. If you decrease the light level, they tend to respond fairly quickly, but if you were to go from 1200 to 1500, you could be waiting 15 minutes for that plant to fully respond to that. And that's not a survey measurement anymore.
No, you're going to be not only wasting your time, but more than likely you'll cave before the 15-minute
Mm-hmm.
is over and
And
you'll
push the button.
and push the button, right? Light availability, canopy structure, some of these turbulent effects given that structure, temperature and energy exchanges. We talked
Absolutely.
briefly about that. Can you expand on it though?
Yeah. So if your plants are either sunlit or shade, they're going to be experiencing a completely different temperature environment. And temperature is going to have an effect on leaf behavior. We all know that all leaves have a T-opt, an optimal temperature. If they're above or below that, then they're going to respond differently. And especially if they're too far above that, if you get to T-crit, you are going to start having some problems. The
Right.
respiration rate spikes, the fluorescence spikes, your plant is just way too hot. It is doing the human equivalent of just like fanning itself or or trying anyway. So if you have a difference in light level, it can also cause a difference in temperature response. And this also is affected by something called the diurnal cycling.
Right.
So the diurnal cycle is how the plant responds to its environment over the course of a day, a diurnal period. And there's always a spot at which the relationship between PAR and assimilation break, essentially, because you are in a situation with high light, low water availability. You could be dealing with some heat stress, some water stress, some nutrient stress, and so you get what's called the midday depression. And that is specifically one of those things that we have to keep in mind when we're choosing a leaf. Not just which leaf, but when leaf.
Right. Yeah, we've talked a lot about the what and the how in terms of gas exchange, but yeah, not really about the when. So this is something that I heard as a grad student, right? Midday depression is the afternoon nap that not only you want to take, but the plant wants to take.
Exactly. And some plants recover from that midday depression. They'll go back up to a higher photosynthetic rate, not usually as high as it is in the morning when they're really getting going, but, or as the photosynthetic world likes to refer to it, cranking. I don't really know, but
That is very true.
it's so true. If, oh man, that one's cranking. Why did we start using this terminology? I don't know, but I learned it, I taught it, it's just a thing.
I think it has to do with because it's like the machinery, the
Maybe
photosynthetic machinery,
it's
and
cranking along.
maybe.
Maybe, I don't know, but it's so common to have someone say, "Oh man, that one's cranking," you know. But it usually does not quite get to that mid-morning level. It's usually a little bit lower, and that has a lot of biochemical reasons that this could be occurring.
Right, right. A lot of it has having to do with that T opt, right? The optical
Exactly.
leaf temperature, and then as well as the VPD
Right.
increase, and those together have this bimodal kind of effect, right? That shows that
Absolutely.
increase or decrease in assimilation and conductance. We'll get back to the podcast right after a word from our sponsor, Sir David Attenborough. In the quiet understory of the forest, we encounter a most remarkable creature. The leaf. To the casual observer, it seems unassuming. Yet, it is here that the very breath of life is forged. Photosynthesis. Approach it with care. Too swift a movement may disturb the delicate balance of its stomata. One must be careful when placing the leaf into the chamber. Seal the edges, ensuring no leaks occur. And then the magic begins. Invisible gases, measured with extraordinary precision, reveal how sunlight fuels this humble architect of our world. Truly a marvel in miniature. We've got talking about midday depression and some of the diurnal tendencies
Mhm.
that are observable. Do you have more that you want to say there?
Well, the one thing to be aware of is that there's kind of a pattern, a midday pattern that we see in textbooks. And a lot of times those textbooks are data from crop plants, for example. And I understand it, crop plants are easy, but you do need to be aware that your plant's diurnal pattern could be very different. So, for example, if you were working with a CAM plant, we think of CAM plants as only taking up CO2 at night, but some CAM plants have some morning assimilation. And so their stomates are open and they're still taking in some CO2 early in the morning. So they're going to have a little bit, they're still coming down from their nighttime assimilation rates, but you're still going to see a little bit and then they're done by about 10 a.m. They're just done. Then there's also some tropical trees that have a tendency to do all of their photosynthetic assimilation in the morning. And after about noon, they just stop.
Yeah.
There's nothing you're going to get out of them if you go out at 2 p.m. You're just, or 2 a.m. actually, either. But you're just not going to get anything out of them if you go out at 2 p.m. because they're done for the day. They've done what they needed to do. And the other aspect is to consider life history. So if you have a crop plant and it has to live its entire life from germination all the way to senescence in 4 months, it's going to have a different philosophy of life than if you have something that lives for 1,000 years, 100 years, whatever. So live fast, die young is true for plants too, and that will affect your diurnal patterning. So don't just think, oh, I saw the diurnal pattern in my textbook, and that's what my plant is going to do. You cannot be sure of that.
Right. I want to go back to the point you made live fast, die young is actually what Elizabeth Harley says on on the
Yes.
back.
Uh-huh.
Yeah.
Totally.
Uh, yeah. So I think you're highlighting a lot of important things, and something that I know you've talked with students and researchers about. And I know that because I've talked with people, and if they're desperate enough to come to me with questions, I know I know that you've already heard them. But that is, trying to figure out how much data one can collect in a photo period,
Yes.
and understanding that midday depression
Yes.
is there, and trying to get as much data as possible up until that point. So
Mm-hmm.
this is something, if you're one of those individuals, if you're someone who is trying to design their experiment, use a little bit of the logistic side of your brain and understanding how long it takes for a single measurement, how long it takes to walk from one plot or individual to another. These are really important things because that will help you avoid this midday depression and have these weird artifacts in your data.
Absolutely. I was at a conference a few years ago, and someone actually said, well, I was hoping to have 3 sites, but then my theory ran into my reality, and so I ended up with 2. And I really appreciated that she mentioned that
Yeah.
she had wanted to have more sites, but realistically, you've got to get in, you've got to get out. It goes back to the original statement
Mm,
of we just cannot measure every leaf on the plant or every leaf in our study species grove, if it's a grove of trees. We just can't do it.
sure.
Therefore, choosing the leaf is really important, and being aware of time of day and light level and temperature and all of these things so that the data that you get is as useful as possible.
100% agree here. One of my favorite lines in the book. First of all, this book has one of my favorite illustrations, so that we'll get to that a little bit later. But one of my favorite lines in this book, at least this chapter, is it is important to note that plants are not instantaneously brought into existence as the whole organism observed at any one time.
Poof!
And it's yeah, poof. It's kind of like a no duh thing, but at the same time, like, people at different ages respond differently, function differently. And so, like this impact of age, what is the impact of age on the observable physiology?
Oh, so this is one of my favorite things to talk about because, as I mentioned, I came out of a carbon source sink relationships lab in graduate school. So we did a lot of talking about and thinking about whether different aspects of the plant were carbon sources or carbon sinks.
Mm,
So a carbon source means that it's putting more assimilates into the overall plant organism. A carbon sink means that it's taking more assimilates from the overall plant organism. A young, still growing leaf tends to take more resources than it provides, and a senescing leaf tends to take more resources than it provides. But if you have a young, sunlit, fully expanded leaf, that leaf is likely to be a carbon source. And that's probably the one you want to think about measuring if you don't have another reason. Maybe you do want to look at the young, non-fully expanded leaf. Maybe you want to see how that behaves. Maybe you do want to look at the senescing leaf. That's back to your research question. But if you don't have a reason to pick another type of leaf, then you want a young, fully expanded leaf so that it is a carbon source for the plant and you will have a higher photosynthetic rate. The highest photosynthetic rates for that particular plant are likely to be in those young, fully expanded, sunlit leaves.
right. And so I'm very impressed by the answer because she actually got through the whole thing without saying it depends.
I did.
That was That was really impressive. I did see like a vein pop up on her forehead, like trying to
Yeah.
really struggling to not say it. But the age,
I did say, though, that it was related to your research question, so we could say that that was a restatement
hey,
of it depends.
we're not gonna split hairs.
Okay.
Let's not do that here. But yeah, the age of the leaf and is inherently or can be inherently linked to the position. So there's another like a cascade of this
Absolutely.
inherent link, right? So some growth forms,
Hmm.
anything from a shrub to an annual grass.
Right. And the other extra fun detail is that age can have a major effect on even the type of photosynthetic mechanism that is active. There's a particular genus of tree that at one age is doing one kind of assimilation and at another age is doing another kind of assimilation.
Ugh.
So imagine being the person who was trying to measure that particular. I think you'd have to ask Jason the botanist, and he would know exactly which one it is. But there is one particular genus of tree that on the same tree branch, you could have two different ways of taking up carbon in terms of C3, C4, CAM.
Oh, boy.
That just makes life hard for poor defenseless graduate students.
Very interesting. Would not be something I would want to tackle first.
No. No, no, no.
Probably avoid that one. I know that you are a crocheter. Is
I
that
am.
what?
Yes. That's a good way to say it.
And so I have crochet questions, but you can see this is really good podcasting. Look at this. Questions is started with a C
Okay.
to match crochet.
Crochet
I thought that was pretty silly. So I once again consulted ChatGPT. This is not something I often do, but I asked, because I don't know how to crochet, but I did ask ChatGPT if it was aware of any famous crocheters.
Okay.
It definitely misinterpreted, or I had a bad prompt, in that it gave me names of famous people who crochet, not famous crocheters,
Okay.
which is still neat anyway. But I'm going to read some names, and if you would maybe pick who you would like to crochet with. Now,
Okay.
I don't know your style, if it is like, going with someone's grandfather fishing, in which you sit in silence and that's wonderful, or if it is a more conversational social hour sort of thing. So I've got some names here,
Okay.
and if you don't know who they are, that's fine. I'll read who they are.
Okay.
So Tom Daly is a Olympic gold medal diver.
Okay.
Michelle Obama,
Okay.
Rihanna,
Okay.
Kate Middleton,
Okay.
or Meryl Streep.
I'm picking Michelle Obama because I read her book, her biography, a few years ago. And because everybody was reading it, it came out and everybody was reading it. I don't remember the title of it, but it was interesting to see how her life had gone. And I think she would be the most interesting. I'm not a music or an acting person, so, and I have no interest in whoever Tom Daly or Kate Middleton is, but I know who Michelle Obama is.
So, okay, fair enough. If I was a betting man, that's who I would have probably picked. Although, you never know, Rihanna could have made it on there. The final part of this chapter gets into kind of a unique hydraulics, water specific type of phenomena. And it also includes my favorite. I was telling Elizabeth this earlier. It gets into my favorite illustration, at least of this chapter, maybe of the book. Like, it's pretty fantastic. It has to do with measuring attached versus detached samples. And so, like, I did my research in a grassland. And so, like, top canopy was not really a concern for me. I'm also not very tall, so this was like the ideal. It was either grasses or like lichens, right? I didn't have a lot of options. But accessing the top canopy, so those most fully expanded, sun-fed, mature leaves, right, are typically not accessible from the ground.
No.
In a lot of systems.
Yeah,
and
that's
so
true.
What are folks supposed to do about this?
So there's a couple of different techniques that people will use to get them onto the ground, and those are always fun to talk about because they often involve literally shooting branches out of trees.
Yes.
You have to get very specific permits for these things. But the other thing that people do is they'll climb the tree and they'll cut it down with a saw or something like that. The challenge with any of these measurements is being sure that the plant responds detached in the same way that it would attached. And if you're going to do these measurements, you really have to be careful of that because, I mean, you may say, well, I don't have another option. I can't get to the top of my canopy. I have to shoot it down. And that's fair. But you do need to be aware that as soon as you detach the leaf or the branch or the limb from the larger tree, you're going to have a hydraulic response. Essentially, the hydraulic flow inside of the leaf is a constant stream. And then as soon as you do that, it simply goes pop and it just splits off. And it almost kind of bounces back into the cut ends in terms of the water behavior. Now,
Mm-hmm.
I am not a water potential person. So if any of our listeners are water potential people and are listening to this and going, that is not how it works, I apologize. That's my understanding of it, is that, when you break that stream, that they kind of pop and they kind of release back in. And then if you then put that branch underwater without doing anything else, you've still got that little bubble.
Right.
So what you have to do is recut underwater, at which point, oh, okay, we can reestablish the flow of the nutrients and the water into the leaf. And so it'll last a lot longer.
Right.
But if you don't, then you're going to find that your plant just shuts down. And how long it takes to shut down? Well, it's my favorite t-shirt again. It depends.
Yes.
The more, I mean, if you cut a little 6-inch branch, it's going to shut down almost immediately. However, if you did a 6-foot branch, a 2-meter limb, you're probably going to get at least a little while out of that, but you got to prove it.
Right. And that's something that is emphasized in this
Mhm.
chapter, right? Is making sure to validate
Right.
these measurements, because there are certainly those examples in the literature that report this. And I've read some, and you've probably read some too, where you're not quite certain how they did it. Right.
Yes.
So first of all, one should explain that method,
Yes.
because it depends. Gosh darn it! I made it so far into this episode without doing that on accident, but it really does. And so if you are one that needs to do that, then validating that, and it's going to be species dependent. It's going to
Absolutely.
be ecosystem dependent, life stage. I mean, there's myriad reasons that you would see a certain response.
Yeah, I was talking to someone recently, and they were only, when they were concerned that they were seeing a very steep drop in stomatal conductance as soon as they put the leaf into the chamber. And so we started talking about, okay, what were your environmental variables, and what's a little bit more about your research question, and what's your sample, and what's your species, and all of that good stuff. It turned out that they were cutting 6 inches. And they had a couple of leaves. I think they were working with oak trees. They had a couple of leaves on that 6-inch branch, but they just didn't have a lot of reserves in that small amount of tissue in that stem. Whereas if they were to, so then they were going to try, I said, I don't do experimental design, but you might try a longer branch and seeing, because it's got more reserves that it can pull from in that case. And then you might find that things work out better for you. But you do have to prove it. You have to show to reviewer number 2
Oh,
that you know that there could be a detachment response, and you've got to do that. I'm not a big fan of the fact that materials and methods are now in tiny, tiny print or in these supplemental materials of papers these days. I understand you want to talk about the pretty cool graphs and the discussion and everything like that, but I really want to see your materials and methods so that I can understand if you did consider detached versus attached. And fluorescence, which this is not my chapter, but if you were going to do dark adapted fluorescence measurements and you were going to do a 30-minute aluminum foil thing, you have to make sure that that matches your pre-dawn measurements, or else reviewer number 2 is going to have questions for you. Are you sure that that actually is a comparable measurement?
100% agree. Elizabeth, go and step down from the soapbox.
Sorry.
There you go. There you go. No, I loved it because I agree. If reviewer number 2, they should be asking that.
Yes.
That's how I should say. Reviewer 2
So
should be asking these types of
I
questions.
try not to be reviewer number 2, but I ask those things. I don't get asked to review very often because I don't have a PhD, but I have reviewed scientific papers, and if you don't put those kind of details in there, I'm going to have questions for you because I know what you should have in there.
Normally I call this the wrap up, but today I'm calling it the fasten off.
Okay, very nice, very
Yes,
nice.
thank you.
Yes.
And so I asked this of every guest. If we didn't record the previous 40 odd minutes, what are some key takeaways? If you were just to give a couple key takeaways that you would recommend for people to really fixate on in this chapter, as well as exploring further, what would they be?
Know your leaf. Know your leaf in terms of what is its age, what is its stage, what is its time of day, what is its growth habit. Know your leaf, because if you don't know your leaf and you just randomly pick a leaf, you might get good data, but you might not. And data is not very good at being Play-Doh. You can't usually take 8 hours of data and then squish it into the form that you want it to have. So if you think about your leaf and you know your leaf beforehand, it will save you a lot of trouble afterwards in terms of, that's 8 hours of my life I'm never getting back. Now that's going to happen to everyone. we will all take data and go, well, I'm never using that. But let's try to minimize that. So know your leaf.
Very well said. Thank you so much, Elizabeth, for joining me here today. I know that you're very busy, and I appreciate the time taken from your day and coming and talking with all of the viewers. And thank you, everyone who is listening.
Absolutely. I'm very glad to do it. And I will once again say, if you have not contacted our tech support team and you have questions, just contact us. People say things like, oh, I don't want to bother you, or my question isn't very good, or I should be able to figure this out myself. Just call us or email us. We are here to help. It is what we like doing. It is literally my job, and I love doing it. So please call me and help me do it.
Thank you, Elizabeth. Please remember to like and subscribe so you don't miss any episodes. And please remember to thank our incredible science and support group. They are, in fact, some of my heroes. And as always, stay curious, stay kind, and I'll talk to you soon.